So let's get started. And I'd like to say welcome to everybody to this lecture of part of the Research Festival. And it's a pleasure to introduce Professor Felicity Gavins, who's going to give us the talk. And before I introduce Felicity, I'd like you to think about questions you might want to ask. We have the Q&A function, so please put your questions into there. And at the end, we'll have an opportunity for me to ask those questions of Felicity. There's also the chat function as well, so please feel free to put things in there as well. Now, let me introduce Felicity. So Felicity read pharmacology at the University of Sunderland, and then did a PhD in pharmacology at Queen Mary University in London, supported by the British Heart Foundation. Felicity was then awarded a British Heart Foundation Junior Research Fellowship to undertake further research in the UK and the USA. In 2007, Felicity joined Imperial College London to take up a lectureship position in the Centre for Integrative Mammalian Physiology and Pharmacology. And this was shortly followed by a senior lectureship and the appointment to the deputy head of the Centre for Neurodegeneration and Neuroinflammation. In 2013, she went across the pond and she accepted an academic position in the USA at Louisiana State University Health Sciences Center in Shreveport. And I think she'll be talking about that in a presentation. I think there's a picture of that institution as well. And she was also appointed director of the small animal imaging facility there. And then we were very pleased to appoint Felicity to Brunel in August 2019 as Professor of Pharmacology, and she was also received a Royal Society Wolfson Fellowship, a very prestigious thing for her to have done, and she's also Director of the Centre for Inflammation Research and Translational Medicine. Her research aims to reduce inflammation in the brain following a stroke in order to limit cell death and contribute towards an improved recovery. Felicity, we look forward to your presentation on Jekyll and Hyde, the monstrous side of inflammation.
Thanks, Paul. Thanks for the introduction, and welcome everybody. It's rather a strange event to have, you know, this format when you can't actually see people. So, but I'm very pleased that everybody's joined this evening. So I'm going to take you through a bit of a journey, really. Paul's already said about a little bit of my background, how I started, where I did my PhD, etc. So I'm going to talk a little bit of history, a bit of history about inflammation, and really how it's led me to the path that I'm on today and the research that's being undertaken in my lab at the moment. So you can see here on the right hand side of this, the initial slide, 4 different institutions. Paul's already mentioned them, but really they've been, I've been very fortunate to be able to work in different places. And each one has provided me with a rich environment in which to flourish doing research in different areas, I would say. And currently, as you can see, I'm at Brunel University in London, which I joined towards the end of 2019. Okay, so the outline of the talk, really, as I say, I'm going to talk about inflammation. I'm going to discuss why I've said Dr. Jekyll and Mr. Hyde in the title. If you don't mind indulging me, I'm going to also go through a bit of history about the inflammation, and history in the context of where my research is going today and how I've got there, I suppose. We'll talk about this aspect of resolution of inflammation, what it is, what it means, and why it's important. And then I'll put it in the context of some of the findings that we've made and that we're doing at the moment, the research strategies that we're doing or undertaking at the moment. And then I can't not mention, obviously, COVID-19 in the context of inflammation, and also I'll touch on thrombosis in the context of that of the virus. So why Dr. Jekyll and Mr. Hyde? Well, you can see here, this is the cover, the title page of the first London edition of the book of The Strange Case of Dr. Jekyll and Mr. Hyde. And it's the reason why I chose it is because inflammation is both a very positive thing and a normal response of the body to ward off infection and pathogens and foreign substances that might present themselves to our bodies. So under normal circumstances, that our body is fantastic in being able to cope with those situations. But it also has a very sinister side as well. And it has a side where if it's not controlled, it can go really out of control, you know, where you get this chronic inflammation and these mediators that are involved in that. And as we go through the talk, I'll share more of that information with you. And as Paul said, more than happy to answer questions at the end, so please feel free to ask. So inflammation, what is it? Well, if we look at the term inflammation in the dictionary, it comes up with a localized physical condition in which part of the body becomes reddened, swollen, hot, often painful, especially as a reaction to injury or infection. And I'm sure many of you have seen this schematic before. It shows that what we class as the 5 cardinal signs of inflammation, and those are heat, redness, swelling, pain, and loss of function. And those signs of inflammation really date back to the 5th century BC. And I really want to take us right back there and then start to explore where we are today in the term of inflammation and what that means. So let's take a walk through history in the context of obviously my research. So Hippocrates, 5th century BC, that's where we're at. Hippocrates was a Greek physician. He is recognized as the father of medicine, and that really is because at the time diseases and illnesses were thought to be sort of omens and bad events that were occurring to you for whatever reason. And whereas Hippocrates realized that actually these are real events that happen, and he recognized that inflammation is an early response to healing. And so because of that, he's really coined the term, I suppose, father of medicine. A lot of the terms that he used then, such as edema and sepsis, we still use today in medicine. And I've put in the bottom there about aspirin, and it's a little story, and we'll touch on this as we go through as well. But the active ingredient of aspirin is salicin, and that comes from willow or willow bark. And so Hippocrates is often, throughout history really, has been linked with what we now know to be the drug aspirin, but mainly the salicin side of it. And he's been linked with that, and it's been thought or suggested that perhaps he'd said to his patients, you know, to drink tea from the willow bark or to chew the willow bark in order for medicinal purposes. But when you actually look through the history, there's only really one definition where he's mentioned, where he's discussed it, where he's written this down, and that's where he's burnt the willow bark and he's sort of fumigated the uterus for healing purposes in those women that have had miscarriage. And so for healing purposes, that's what is used, you know. We know that Bayer patented aspirin in 1899. And the reason why it's sort of interesting for me, not just because of Hippocrates and starting off there, but also because when Paul mentioned that I did pharmacology as my undergraduate degree, but during my second and final year, I actually did an industrial placement year, and that was at Bayer Pharmaceuticals, which was in Stoke Poges, which is just down the road from Brunel. So it feels like a bit of a circle. I've come all the way around. Bayer's no longer there, but it was instrumental because the gentleman I worked for at the time was a gentleman called Dr. Vince Russell. And the area of research that we were on, it was all about lung and respiratory diseases at that particular site. And I was focused on isolating neutrophils. These are white blood cells in our blood, isolating neutrophils and looking at their function by stimulating them or activating them, or humanized neutrophils, activating them, and then using or targeting them with different drugs that were in the Bayer repository of drugs that they had for the different, looking at the different responses in there. So that's really where my fascination with the neutrophil suddenly started, and with inflammation. And this will go through as we move through the talk. I'll discuss this more and more. Interestingly enough, Paul's already said that I worked at the William Harvey Research Institute, and it was at this institute where Sir John Vane worked. And it was Sir John who was able to find the mechanism of action for aspirin. And so he was given, or awarded rather, the Nobel Prize for that work, looking at the mechanism of aspirin, the drug that we use today, that so many of us use today in the context of cardiovascular diseases, for example. And in actual fact, that when I did my PhD, my viva, I actually had my viva in Sir John's office. So this whole roundabout way, I suppose, of not just the 5th century, but it moves through and seems to have touched pretty much every aspect of my research. So if we then move to, so I've shown you already these 5 cardinal signs of inflammation, and certainly the history of immunity and inflammation is very long, and it's very interconnected and entwined. And I'm only going to spend a little bit of time on this, as I say, in the context of what I do and the research that I perform. So it was Celsus who came up with the 4 cardinal signs, and it's known as the Celsus Tetrad of inflammation, and those being the heat, redness, swelling and pain, or the calla, ruba, tumor and dolor. So he coined these terms, and he saw those terms because he saw inflammation when he looked at surgical procedures that were done, and I've listed some of them there, that he saw that inflammation was also associated in some cases with those procedures. Which obviously we don't want it to be, but some infection, etc., occurred, and so he saw them there. So he coined the four terms. It was Galen shortly after that came up with the 5th sign of inflammation, this loss of function. And obviously we don't always have loss of function. We tend to have the 4 cardinal signs, but not always loss of function. But in some diseases and disease states we do, such as rheumatoid arthritis, we have that loss of function that can be there. But Galen was a Greek physician, he was a surgeon, and like many physicians at the time, also a philosopher. And he was a very well-respected and renowned surgeon. He was also the physician for the Roman emperor at the time, Marcus Aurelius, and also his son. So he was very well respected. And he was also respected because perhaps they didn't necessarily recognize it then, but he was instrumental in understanding that the body produces certain responses or certain substances in response to certain events. So, for example, if we eat something that's hot, then in some cases we'll produce this yellow bile, this yellow substance. And so they always described at the time the fire within. And whereas it was Galen who really realized that it's an actual event that's happening in the body to produce these responses in connection with something. And so he really sort of coined those terminologies, although perhaps he didn't know what it was. Obviously now we know that it's helping with digestion, we know the events that occur, but at the time, he was the one that first started with that. I can't not mention William Harvey, obviously, having worked in the Research Institute named after him, and in conjunction with St Bartholomew's Hospital. And I would say when we come out of the pandemic, I would recommend that if you can, to take a visit to the Harvey and to St Bart's and have a look at some of the history that's there about William Harvey. He was an English physician, and he was the first one to describe a circulation and the fact that it's the heart that pumps the blood to the rest of the body. It's of particular importance to me and obviously to everybody really, but for my research, because not only because I was at William Harvey, but because I'm interested in the circulation, and in particular the microcirculation, so the very small vessels within our circulatory system. So not just the larger vessels, the arteries and veins, but the arterioles are smaller vessels of that, and the venules as well, because this is where inflammatory and inflammation starts. And so that's where, if we can suppress that or control it at that point before it gets to the larger vessels, then perhaps we'll have a better end result in certain situations. So it was William Harvey who first described the circulation, and he's got some fantastic experiments that he did in order to see which way the blood was pumping, looking at the forearm, pressing the vein in the forearm, and then looking at the way in which the blood was flowing and things like that. So it's fascinating techniques, and some of those techniques we still use today, although perhaps we've modernized them slightly. With all of inflammation, one of the techniques that we use to quantify inflammation and to literally observe it on microscopes, and we use, certainly in my research, we use some very fine microscope systems. And so I can't not really mention two gentlemen that were instrumental in describing and characterizing and formulating and inventing really the microscope. And the first one is Janssen in the late 16th century, who invented the compound microscope, and that's a picture of it there. It's quite simple really, I suppose, but obviously not at the time. So you've got the eyepiece, you've got the objective, and so it's the first time that obviously you could start to see, to objectify things and observe events. But it was Leeuwenhoek, who's described as the father of microbiology, who was the one really, the first person to make or invent a single cell, invent a microscope, an optical microscope that was really able to increase the resolution, and by that being able to observe organisms, single cell organisms. And that's one of the reasons why he is coined the father of microbiology. And this is a picture of his little microscope. And I say little because it really is about the size of your thumb, so it's not big at all. It's got a couple of parts here, little bits to hold the specimen in place, which would be held here, and then a tiny little objective, and so you hold it up to your eye and you can see these single cell organisms. But he really was instrumental in setting out being able to visualize cellular events. And the last few gentlemen I wanted to mention, one of which is this gentleman here, Rudolf Verkoff, who was a very distinguished and experienced German physician. He had many talents as well. But the reason why I wanted to bring him to the forefront, I guess, is because he was the first to describe diseases, particularly diseases within the blood and events in the blood, such as embolism and thrombosis. So the idea of clotting that occurs in the blood in disease states. And we use this sort of Verkoff's triad nowadays as well in our research. You know, the three elements that are so important for thrombosis to occur, in particular venous thrombosis to occur. So the status of the blood flow, endothelial injury, and by that we mean the blood vessel wall is lined with endothelial cells, and it's the injury to those cells that's part of how thrombosis occurs. And we also need this hypercoagulability, this increase in clotting that occurs, and those three elements contribute to thrombosis. Now those elements, and it being termed Verkoff's triad, actually is in a way nothing to do with Verkoff. It was coined, those elements were defined and coined after Verkoff had died. But it's because he was so instrumental in setting the groundwork and the foundations that his name is associated with those elements, because of the work that he was instrumental in doing. So the final two gentlemen really that set the scene for my research are Frederick Recklingshausen and Julius Cohenheim, both of whom were German pathologists. Julius worked in Frederick's lab, and Frederick really characterized pus cells in acute inflammation, and that's the sort of yellow substance, I guess, that you see. And I'm going to show you a picture in a minute, although I'm sure you've all seen it before, or seen sort of, you know, spots and things like that. So that's cellular debris that's in there. But he showed that cells could migrate, they could move and contribute to what we term phagocytosis, or the eating of this, of dead cells, of eating of debris and things like that. And so phagocytosis looks a little bit like this for those of you that perhaps haven't seen it before. These are human macrophage. Sorry, let me just get it going. Yep. Human macrophage here. And these are little zymosan particles. These are just foreign substances, if you like. And you can see that the macrophage is eating those foreign substances, it's digesting those, and that's what happens in inflammation. We, under normal circumstances, these macrophages, and neutrophils can do it as well, they phagocytose these foreign substances in order for us to clear that those effects. But it was Cohenheim who looked at the causes of inflammation. He was the one who used frog's tongues and showed that leukocytes pass through the walls into tissues. And his work was instrumental in what we look at today. And we still do very similar things in today's research, and certainly within my lab, we don't use frogs or frog's tongues, but we use similar techniques to look at leukocytes, in particular neutrophil trafficking. So all of these gentlemen that I've mentioned have been pioneers in their own field and set the scene really for the type of research that I do today. They've all looked at the Dr. Jekyll, the positive side of inflammation. They've also looked at the Mr. Hyde, the negative side, or the detrimental, the evil side of inflammation as well. Although they might not have known what we know today, they are pioneers in characterizing that. Okay, so I've spoken a little bit, so you've indulged me in the history of inflammation in the context of my own research. So now I'm going to go in a little bit more detail about what inflammation is and what we now know today about inflammation. So I've shown you the 5 cardinal signs of inflammation. This is where acute inflammation happens. I've got some pictures here. We've got some skin infections. We've got a nice and growing toenail. There's that pus that I was talking about, you know, that yellow substance that's in the eye because of an infection that's there. That's actually my hand. I had a little piece of glass that was in my hand, and you can see there's that redness, definitely the heat, a bit of swelling there, and some pain, no loss of function, but definitely the 4 cardinal signs. And a couple of days later, all fine, no problems. The body's able to restore it. My immune response was such that homeostasis is returned to normal. And the reason why I had glass in my hand is because I make glass artwork, and so I have a neutrophil behind me who's made out of glass, and so that's part of the reason why it was there. But I took a shot of it because I thought it was quite rather ironic in a way. But normally the analogy that we give is a bee sting. So a lot of people are obviously, unfortunately, been stung by bees, but it's a classic sign of, under normal circumstances, that you'll get those 4 cardinal signs of inflammation. And then over a few days, it will resolve and it'll disappear, and that's fine. However, so when we first started looking at inflammation, or when I first started looking at inflammation anyway, which is quite a few years ago, we thought at the time, from a pharmacological point of view, so from a drug point of view, that the ideal drug candidate would be to get something that you get these, what we call pro-inflammatory mediators. Those are mediators that increase inflammation, that occur when inflammatory environment is around. So we thought that the best idea, and say we, the field, thought that the best idea was to find drugs that suppress those pro-inflammatory mediators and perhaps increase anti-inflammatory mediators in order to restore that balance. We now know that it's not actually, I wouldn't even say as simple as that, but it's that the body is a fascinating paradigm, that it has the way in which it controls different events is really quite remarkable. So it's not as simplistic as that. So we know that you have this onset phase of inflammation, there's a balance response there, and then you have this, what we term resolution. So in other words, restoring the body to homeostasis, to normality. That's what normally happens. The problem arises when we've got chronic inflammation, when we've got an excessive inflammatory response, where the trigger of turning that inflammation, those signals, those events that are occurring in the inflammatory stimuli and environment aren't turned off. And so you have chronic inflammation, and because of that, it's non-resolving. And so the body isn't able to restore homeostasis. So my lab's interested in being able to pharmacologically, so with drugs, understand how these mechanisms are happening in different diseases, but also whether we can treat them with drugs to manipulate that profile and then restore or drive resolution and restore homeostasis. Now, I've said already mentioned already about the cardinal signs. This is of inflammation. That's the onset phase that occurs. And we now know that there is that resolution. I've mentioned the resolution phase. And this, the work in the resolution field has been absolutely pioneered by a gentleman called Charles Sirhan, who's at Boston, who's at Harvard University in Boston, and he has been the pioneer of the resolution field. And so he's really set the scene for saying that it's not simply upregulation of one, downregulation of another. It really is about different mediators that are controlling those different events at different times. Okay, so we know that we get different cells that are involved, that we get the edema, we get the swelling that occurs in the inflammation, and then we get neutrophils or PMNs, polymorphin nuclear cells. Neutrophils are white blood cells that come in. And then after that, we've got the monocytes and the macrophages. This is all innate immunity. We've got monocytes, macrophages that come in that really steal the show when it comes to resolution, because they're the ones that do the vast majority of the work. But I'm the driver of the neutrophil. So I'm the one that's supporting the neutrophil, because although neutrophils can become the Mr. Hyde, they can produce so many inflammatory mediators that create that excessive inflammatory response. They also are the signal when they're right. They're the signal that drives resolution of inflammation. So they're the key cell that triggers the resolution profile. So they play a very strong role in this event. So resolution of inflammation, it's an active process. It's spatially and temporally regulated. So what I mean by that is there are different mediators at different times doing different things, and they're all working together in a very fluid way in order to drive resolution. And the key thing is for us to understand what they're doing, when they're doing it, why they're doing it, so that we can, using drugs and treatments, be able to modulate that in a disease setting. That's what we're interested in. So that's what we do in a nutshell. That is what we do in my lab. And so there's different ways of looking at information. I've said Dr. Jekyll, Mr. Hyde. You could say the Janus, this image that's often put above doors, you know, the entrance and exit of things, the two sides of a coin. So that's inflammation. There's the positives and the negatives. There's a lot of chronic inflammatory diseases out there. I've listed a few here. Some of them you will be familiar with anyway. Those in red are the ones that we really look at in the context of my lab. And in particular, Paul has mentioned about stroke. We've had a lot of work we've focused on in the context of the brain, a very fascinating organ. To measure inflammation, multiple ways we can do that. From the clinic, all of you, I'm sure, have given blood. You've all had a blood sample taken. You know, you've got different parameters that are measured in the blood. It might be cell counts, neutrophil counts, platelet counts. It might be certain markers in there that look at inflammation. It might be markers of looking at how organs are functioning and whether they're having problems, etc. So there's lots of ways of us looking or measuring inflammatory events. We use a technique called flow cytometry that can enable us to look at markers on the surface of the cell, you know, to see whether they're up or down regulated in different events. We can look at what they're producing in different situations. We can take tissue samples and we can stain them for different cells, for leukocytes such as neutrophils, platelets, etc. And we can also do live cell imaging. So there's lots of ways we can measure inflammation. I've already said that I'm interested in the microcirculation, and the reason for that is because that's where inflammation starts off. That's where the events occur. There's lots of events are happening in the venules and the arterioles in the body. And so you can see here in the top, it's a busy slide, I suppose, in some respects, but showing more or less the same thing. So in the top of the slide, you see two staining techniques showing more or less the same thing. You might argue that one looks slightly prettier than the other, perhaps, but it shows the same thing. So it's showing that neutrophils are in the vessel wall in a tissue, and that's an inflamed tissue. It's showing what we call microvascular dysfunction, that those cells are there. And you can see the accumulation of those neutrophils in that vessel. You can see them collecting in that vessel, that microvascular dysfunction. We're interested in looking at how those cells roll along the endothelium, how they move along there, move into the tissue, and the different mediators that respond and cause those effects in different vascular beds. And some are very vascular bed specific. So for example, some things that happen in the liver might not be the same as that happens in the brain. And that's just the nature of those different organs and the functions that they perform. But again, we're going back to Julius Cohnheim, we're going back to the 1800s. He's the gentleman that set the scene for the research that we do in my lab. He used frog stongs to look at leukocytes trafficking. We now use mouse models and other models, zebrafish, for example, models, to look at cells trafficking and understanding why cells move the way they do and how they present themselves and in different diseases, in order for us to be able to pharmacologically treat different disease states. So I wanted to come up with a quick analogy to perhaps set the scene rather than me just simply saying neutrophils rolling along the endothelium. And it was my postdoc some years ago, Helen Smith, who came up with this analogy, which I subsequently realised perhaps wasn't the best thing, given the fact that nobody can really travel at the moment. But nonetheless, so she came up with the idea that neutrophils, or leukocytes, neutrophil is a type of leukocyte, that leukocytes are like planes. And they're coming in to land in the runway, and the runway, if you imagine it, is the endothelial cell surface. So it's these endothelial cells lining the vessel wall, and along the vessel wall, and obviously the vessel wall is cylindrical, so it's not quite the same as a runway, but along the vessel wall you've got these adhesion molecules and different molecules that are there, just like the lights on the runway strip guiding the plane in. It's the same really for neutrophils. There's mediators there that are guiding those cells in so that they're going to the site of inflammation. So they're responding to the body's call to say, there's something here that we don't recognize, come and help, that sort of thing. So that's what they're doing. And that's, I think, quite a nice analogy, really. So we can visualize this in different ways. Again, building on the history that I spoke about originally, we can visualize it in different ways. We can grow or culture endothelial cells, and we can culture endothelial cells from isolated from human cells, or we can do it from rodents and other species. We can culture them, and then we can flow different cells across them. So we can flow neutrophils, for example. We can activate the endothelial cells so that we're more or less mimicking or trying to mimic what we see in the clinic, because every aspect of our work has to have that translational element to it, because we are about trying to get drugs into the clinic that are more effective to treat diseases. So we need to mimic what goes on. It is a model at the end of the day, though, but we are modeling certain aspects of what happens in that inflammatory event. So what it looks like, for those that perhaps haven't seen this before, and I apologise, it might be a little bit small, but what I want you to see is, hopefully you can see these little very white dots are neutrophils, and some of them will be sticking on the vessel wall, and some of them are just flowing through, and some of them are rolling along. And it's this paradigm that's up here, these different events that occur when the neutrophils are interacting with the vessel wall. And we can look at those different mediators and responses. We can treat the cells, and then we can see whether it could be a potential treatment for a particular disease. We can also do this in vivo, and by that we use specific imaging modalities to do that. So one classic one that we use in my lab is intravital microscopy. This enables us to visualize in real time, in anaesthetized animals, cellular interactions in different tissues, in different vascular beds. Okay. So I'm going to show you, this is a mouse, this is a vessels from a mouse that's been initially, there's no inflammation, this is a physiological response. And then after that, I'll show you what happens when we inflame, when we cause inflammation in the brain and how cells respond. We've labelled platelets, and platelets are these cells here that are shown in red. That's a platelet, an example of a platelet. They're the cells that are involved in clotting, in hemostasis. So when you have a cut, they're the cells that are there to help with that clotting, which under normal circumstances is great, but when it becomes problematic and we get thrombosis, as going back to Verkoff's triad, then it's an issue. But those cells are involved in that. We also know that they're involved in inflammation now, which is a relatively recent thing. So the field is moving. So we've got platelets labelled in green, and we've also got neutrophils that are labelled in red. So what I want you really to appreciate is, and remember, this is real time. This is happening in the brain of a mouse. You can see there's nothing really going on within that vessel. And then we come to it when we inflame the vessel, when we inflame the mouse, you can see hopefully that this interaction of cells. You can see these red cells sticking, some rolling, some little green cells as well. That's inflammation. That's occurring in the brain in real time. And we're able to look at those events and modulate them with drug interactions and understand what's going on in diseases. I should mention at this point that you can obviously see that some of the models that we use animal models, so in vivo models, and we also use human tissues and samples And so both of those aspects have ethical implications to them. And so we don't undertake any work that's not, that obviously is well regulated. From the in vivo point of view, it's home office controlled, it's home office regulated. And so we're very, very conscious of the ethical implications of the types of models that we do and the research that's undertaken in the lab. And I'm more than happy to talk about that, but it's important to mention those things. We also work very closely with the NC3Rs, which is this National Centre for the Replacement, Refinement and Reduction of Animals in Research. And that's because we want to be able to get as much information as possible in different settings before we go into in vivo settings and before we go into the clinic. It's very important that we do those simultaneously, but also in a very controlled fashion. Right, so the need for resolution pharmacology really is that I've said that my lab looks at this idea of being able to manipulate this profile. So understanding physiology, the pathophysiology, the mechanisms and the pathways and mediators that are involved in these different diseases and different inflammatory diseases. What we hope to be able to do with that is to then be able to get or obtain better therapeutic targets that are more specific for each individual, that are more or less tailor-made for every person with a different disease. They're very specific, and so being specific, they're very accurate with fewer side effects. So what we're looking at really is looking at what the body does under normal circumstances. I've said about the acute inflammation, I've said about the resolution and the homeostasis and the restoring to homeostasis. So we're interested in what does the body do? Normally it does it very well. So if we could help the body do that, and so look at those mechanisms that go on, where it's the normal body response to an infection. If we can modulate that and characterize it and create a drug for that, then surely that's better long term, because the body is more adapted to accepting that as a drug, and hopefully that drug drugs have fewer side effects. So as I say, we take a multidisciplinary approach in my lab, using different models, including sort of, as I say, rodents and zebrafish as well. So having sort of explained what we do and in the inflammation side of things, I can't not really mention a little bit of data and research and findings in the context of one of the drugs that we've been interested in over the years, and that's annexin-1. It's a protein, and I started working on annexin when I was doing my PhD. So quite a few years ago now, and we still use it, we still work on it in different ways. But ironically, I guess, again, it's about this full circle, these how things are interconnected that perhaps at the time you don't realize, but as you go through, and certainly putting these slides together, it made me think about how things are so intertwined in my research career that perhaps I didn't think about them at the time. Hindsight's a wonderful thing, I suppose. But anyway, so the annexin protein, it's like a donut. It's the shape of a donut. It's got these 4 conserved repeats, so just 4 areas of it. And inside that donut is this sort of what we call an N-terminal domain. And it's shown there in yellow at the top, and that's the structure of annexin. And this N-terminal domain, because it's a protein, it's full of peptides. And that N-terminal domain is the fingerprint for annexin. So just as we have fingerprints to identify and make us all individual, the annexins in the superfamily of annexins also have their own fingerprints, which is this N-terminal domain. So it's very specific for each one. And these 2 gentlemen that I've shown at the top right-hand side, Rod Flower and Mauro Peretti, were actually my PhD supervisors, and really were pioneers in the annexin field of driving it forward, and instrumental really in driving my research and the curiosity and developing things. So it's certainly thanks to them. So neutrophils, ironically, neutrophils have a lot of annexin in them. We can see that, we can quantify it by labeling the annexin with, in this case, we've used again a microscopy approach. We've used what we call an electron microscope, so we can really see in depth into the cell, and we can see when we immunogold label the annexin, you can see these little, hopefully, little black dots within what we call gelatinase granules. So in other words, these little lobes within the neutrophil itself. And that's the annexin. That's where it's stored. So we know it's there. We can see it. We can quantify it. And we know that if we fluorescently tag it, it doesn't matter what sort of fluorescence, green fluorescence, if we fluorescently tag it and we activate the neutrophil, it releases its annexin, because that's its response. That's what it does in order to help drive resolution. It's a positive response. Annexin is an anti-inflammatory mediator. It's quite an unusual mediator. It's anti-inflammatory as well as pro-resolving. Okay, that's what it does. So it releases its annexin, goes onto the cell surface, and then interacts with what we call the formal peptide receptors, or FPRs. Think of the FPRs and annexin like a lock and key. We use this analogy in pharmacology an awful lot. And it's the idea that the FPRs, these receptors, are the lock, and annexin is the key. And annexin is able to unlock the FPR family, and in particular, it's able to unlock FPR2. In the gut, it's another member, FPR1, but typically, in most cases, it's FPR2. And that FPR2 is a key receptor in the resolution profiling. And so annexin is able to unlock is the key to unlock the lock, obviously, and then it causes events in the cell, and it changes. So, for example, it can cause detachment of those neutrophils from the endothelial cell surface. It can instill phagocytosis, that image, the video that I showed you before, so that the neutrophil can engulf, let's say, platelets. It can also be itself engulfed by macrophages in order for clearance to occur. For that clearance is necessary for resolution to happen and homeostasis to occur. And this paper that's at the top right-hand side was a paper that was instrumental and in a way formulated the foundation of my PhD, which was, it was a group from Germany, Volker Goeckel's group in Münster in Germany. And he showed that, or his group showed that annexin-1 mediated its protective effects, so it's anti-inflammatory effects. At the time, we didn't know resolution, but anti-inflammatory effects via an interaction with the FPRs. And he showed this in vitro models. And so part of my PhD really was to say, okay, it's been shown in vitro models, and that's great. That was a real strive forward. But is it the same in a physiological setting? You know, in a disease state, is it the same? And so part of my PhD was to look at that. So again, coming round, we can see that you don't need to, this is quite a busy slide, so you don't really need to know this, other than it's just showing that these are mediators that are involved in resolution profiling. And you can see here that, again, Charles Serhan is pioneered this, but you can see again with aspirin interacts and can form what we call aspirin-triggered lipoxin. And this is a key, an even better key, really, for the FPR2 receptor. So it's more specific perhaps for that receptor. So again, it comes all the way around, full circle in the research that we do. We've shown it as well. Since that, we've shown it in animal models using intravital microscopy, using that methodology that I showed you, you know, with the platelets and the neutrophils rolling in the cerebral circulation in the brain. We can see that if we give animals aspirin, if they've had a stroke, we give them aspirin, we can see that it reduces the number of neutrophil platelet aggregates, and you can see there on that graph. And if we block the lock, so if we block the FPRs with what we call an antagonist, so that's a drug that binds to a receptor but doesn't cause a response, but in other words, it stops that lock from working because it, if you like, it blocks it up, so that annexin or the aspirin, the key can no longer go into the lock because it's stopped. So if we do that, we can see that these neutrophil platelet aggregates are no longer form. And so we know that that response is being mediated through the FPRs. And again, we can look at this in a genetic way and using genetically modified or engineered mice that don't express the FPR2 receptor, that we can see that in those mice, if we give them aspirin, again, there's no response. So it's the FPR2 that's driving those responses. And we looked at downstream signaling of those and characterized that quite a lot. So we looked in particular, we were able to modulate neutrophil function in the context of stroke. So I've mentioned inflammation, and I can't not talk about inflammation in the context of thrombosis. So the reasons for that really are because a lot of the mediators that are involved in inflammation are also involved in thrombosis. And so one sort of begets the other. And so you get this sort of thrombo-inflammatory phenotype or environment that occurs. And in the center of that, I've popped the neutrophil, because the neutrophil is really key in the inflammatory thrombotic field, in this thromboinflammation. And also obviously its interaction with platelets from the thrombosis, from the clotting side of it. But it's the neutrophil that drives a lot of these responses. Now, I've said that we look at different diseases in my lab. Paul's mentioned the fact of stroke in particular. And so I'm going to present just a little bit of data, just to sort of whet the appetite and set the scene of why we do what we do, and some of the findings that we've obtained. So we know that stroke, and I don't want to throw stats at you, but we know that stroke is the 5th leading cause of death in the UK. We know that every 40 seconds somebody has a stroke. And it's the leading cause of adult disability in the UK as well as globally. So it really packs a punch. You know, it really has a huge impact both on the effects on the person that's had the stroke, on their family, on the rehabilitation and the treatment of these patients. It has a huge effect. And really the only treatment for stroke, and this is ischemic stroke I'm talking about, there are different types of stroke, but about 85% of strokes are ischemic in origin. By that I mean that the blood flow is stopped to the brain. So we've got this thrombotic event or inflammatory or thromboinflammation event. So it stops the blood flow going to the brain. And so with any event that's ischemic like that, where the blood flow is stopped, we call that ischemic. You want to reintroduce the blood flow. You want reperfusion to occur. In the context of the brain, you want that quickly, because time is brain. You know, it's important in any other organ, but in the brain, absolutely crucial. And so the two ways of doing that in the context of stroke are either drugs, and there's only one drug that's licensed, and that's tissue plasminogen activator, or TPA, or it's surgically, and that's the removal of the clot. Now, both those obviously have risks associated with them, and certainly in the context of the TPA, you have to give that drug within about 4.5 hours. There's some evidence to suggest 6 hours of the onset of the ischemic stroke. So that means that the person has had to have had a stroke, gone to hospital, identified that it is an ischemic stroke. Because if you give that drug to somebody that hasn't had an ischemic stroke, but they've had a different type of stroke, so one that perhaps is where there's a bleed in the brain, that drug can then increase that bleeding, and that can cause death. So you've got to be absolutely sure that you know what you're doing, and you know the type of stroke that person's had, and you've got to do that within 4.5 hours. That's a very short window. On top of that is also the risk that of those patients that actually have that drug, there's only a very small percentage of them that will actually benefit from it. And so really we need to find alternatives. And in some cases, the surgical, the thrombectomy, is also not appropriate either. So what we're doing, what we're interested in, is looking at resolution of inflammation, of being able to pharmacologically find better treatments and treatment strategies for these patients, irrespective of the type of stroke that they've had, of being able to give a drug early, you know, so maybe the paramedic can give the drug. It doesn't matter whether it's going to be protective in ischemic stroke, but it's not going to be exacerbate if they happen to have had hemorrhagic stroke. And so what we believe is perhaps anti-inflammatory therapies may limit thrombosis, and anti-thrombotic therapies may reduce vascular inflammation. What does that mean? Well, for example, drugs like Annexin-1, maybe because it's anti-inflammatory, and it's also, we've shown it to be anti-thrombotic, maybe that's an ideal candidate for the treatment of stroke. And that's something that we've been looking at. You can see in that schematic all these things that are going on in the vessel wall, that thrombotic occlusion, the neutrophils are activated, platelets, etc. We know when we take samples from our mice that have had a stroke, as well as our patients that have had strokes, when we take blood samples from them and we measure mediators in the blood and look at resolution mediators. So for example, in Annexin-1, you can see there that they're reduced. So there is a dysfunction, a dysregulation of these resolution mediators, not just Annexin, there's other ones that we've measured. We also know that neutrophils are activated in stroke, that they produce lots of different things, including these, what we call extracellular traps or nets. And these nets, under normal circumstances, they're there. They literally are like a fishing net. You can see them in that picture. They throw these fishing nets out. They scoop up the pathogens. They take them back in, and then they ingest them. They phagocytose them. And that's for clearance. That's a good thing. That's a good thing that the body does. But under certain circumstances where there is dysregulation, you get these nets being produced, and they provide a scaffold on which other cells can stick, in particular cells like platelets. And with that becomes more thrombotic occlusion that you can see in the top picture there. And that's also, you can see it in this picture down in the bottom. That's a neutrophil. It's got, we're causing a thrombus in a vessel. We can see that these little yellow dots are actually extracellular DNA that's being produced. So we can look at these in our in vivo models. We can look at these nets being produced. Why they produce what they do when they do, we still don't entirely know. And that's something that we're looking at the moment. So I've said about platelets, I've said about neutrophils, I've said about platelets. We can look at platelets. We know that platelets are also activated in stroke. We can measure things in the blood that shows us that they're activated. So we can look at, for example, thromboxane B2. And we see there that in our stroke model in our mice, we can see that there's an increase in that mediator. And we can look at their ability to aggregate, their ability to stick together. Because that's what they do in a thrombotic event. Platelets stick together and they become very active. And you'll see at the top there, there's a gentleman, Gustav Born, and he's the person that actually invented the machine that measures the aggregation. So he invented the platelet aggregometer, and we still use it today. And so we still use it in hospitals, we still use it in basic research. So we still use the principle of the platelets of this aggregometer. And he was at the William Harvey, unfortunately he's passed away now, but he was at the William Harvey Research Institute, and his office, this is just by the way, his office was actually opposite where the PhD students used to sit. So where I used to sit as a PhD student, and many a day was spent talking about platelets, and in particular interaction with neutrophils. But you sort of look back on things and you think, I wish I'd spent more time talking about them. I wish I'd, capitalized on things a little bit more. But fascinating gentleman, absolutely fascinating. So he invented the platelet aggregometer. Again, very small world. Invented that. We know that we can change the phenotype of platelets. We can see if we administer an Nexin-1, we can reduce platelet activation. You can see that in the measure in the plasma. You can also see that it reduces the ability of those platelets to stick together. So what we found was we could also see this in vivo. We could see it in the context of when we looked, using those intravital microscopy I mentioned before. We could see neutrophils with the little red neutrophils stuck around, surrounded by these platelets. We were able to measure that if we treated mice with a Nexin, mice that had a stroke with a Nexin, we could reduce the ability of these aggregates to form. Okay, so we're reducing the thrombotic tendency. And in particular, think about the fact that when somebody's had a stroke, they're often highly likely to have a second thrombotic event. So we're able to reduce those events. And again, we know that's by FPR2 because we added a different antagonist, a different block of that lock. So in that case, we added this WRW4. So we know it's FPR2 that's mediating those responses. So we're able to show the change in neutrophil function. We can change that phenotype of the neutrophil. We can change the phenotype of the platelet as well. So we found that actually an Nexin-1 had rather an undiscovered phenomenon, really, that it could modulate platelets via a particular reaction inside the platelet when it was able to bind to the platelet cell surface. And this just shows what happens in the platelet. You don't really need to know it, other than the key thing is that we are able to modulate the phenotype of the platelet as well as the neutrophil. What that means is that a Nexin, and I've shown some of the research there in this talk, but a Nexin is able to modulate these different parameters. Think about that in the context of disease. If we're able to modulate what neutrophils and what platelets do, these sort of in thrombo-inflammatory phenotype that we see in diseases at different times, what we could do for patients that are susceptible to these thrombotic events that have chronic inflammation, if we can modulate the positive effects of these cells by giving mediators of resolution. So in my final couple of slides, I just wanted to talk about COVID-19. I can't not talk about it, obviously, because of the status of the situation today, but also because of the importance of inflammation, of thrombosis, and neutrophils and platelets within that. So our immune response that occurs in the infection. So I've spoken about inflammation. I've spoken about thrombosis, I've spoken about the crosstalk between those two, the thromboinflammatory events that occur, as well as the interaction between platelets and neutrophils. They're independent yet interdependent processes that occur. And we know that these events happen in COVID. We know that that patients that have had COVID have all these changes in the phenotype of their cells. We know that the infection, that the virus can infect the endothelial cells. This is a schematic from a recent article that's under review. A schematic showing that the virus can infect the endothelial cells, and with that, it produces all these pro-inflammatory, pro-thrombotic events that we can physically measure. So we know that they're happening. And some of it may also underlie why, for example, elderly patients were more susceptible to the events, as well as, I should say, that aging itself comes with what we call a low-grade inflammation, and it's termed inflammaging. That's there as well. So that could create why those patients were more susceptible to the events of COVID, to the virus. Understanding why these events are happening, understanding the phenotype and the changes in these cells is what's happening at the moment. That's what the research that we're undertaking, as well as other groups globally, because to understand what the virus is doing, we hopefully can get better strategies and treatments for patients, and also thinking about long-term COVID as well, or the effects of long-term COVID. So it's very important that we understand that. We're also part of a consortium with another member. It's a European consortium called the CHOW, and that's with another member of my centre, which is, as Paul said, the Centre of Inflammation Research and Translational Medicine, and that's Dr. Luigi Margiotta Castellucci. So we're involved in a European consortium looking at adverse outcome pathways. So we're looking at the events that occur in COVID-19. We're looking, in particular, we're focusing on the thromboinflammatory components, on the neutrophil platelet interactions, the fact that these cell types independently and interdependently are increased in COVID-19. And we believe that they provide the bridge, really, of a lot of the problems that are occurring in this particular disease state. And by understanding those problems, there's a high potential for pharmacological intervention. So in other words, drug treatments, finding better strategies for treating patients. You can see here that in this view, this is the adult respiratory distress syndrome, or ARDS, which is COVID causes. And you can see then the neutrophil, all of these effects that the neutrophil is producing, these extracellular traps, these nets that I spoke about, all these different parameters, and drugs that have been tried and tested on these different events. So we believe that the neutrophil platelet interaction in particular provides quite a key for possible therapeutic targets, and that's something that we're looking at the moment. So just to conclude, really, I hope that I've shown you that inflammation underpins a number of chronic diseases, including aging. So this inflammaging side of things, that there is a strong crosstalk between inflammation and thrombosis, and that resolution pharmacology perhaps is a better treatment strategy for a number of these chronic inflammatory diseases. And by understanding the pathophysiology, the mechanisms, the pathways in disease, we can get effective therapeutic targets and treatments for precision medicine. So in other words, individualized medicine or personalized medicine approaches that are more suitable for individuals. And so using sort of data integration, the idea of what we call drug repositioning or repurposing, so that's taking drugs that are already on the market for other diseases and seeing whether they would be more suitable, or not necessarily more suitable, but suitable for other types of diseases that perhaps those that they weren't originally set out or have been associated or affiliated with, using that approach, and also finding novel targets. My lab and my research is really about finding better strategies to treat these global problems, these cardiovascular diseases, these thromboinflammatory diseases, of which I listed several previously, of understanding the mechanisms that go on there, of taking a sort of multidisciplinary approach in order to answer those questions, those global challenges that we're all faced with today. That's part of the research that we do in the lab, and it's something that we've continued to do, and hopefully we'll do for quite a long time. So finally, I just want to say thank you, obviously, to the members that have helped with this work. And obviously, this is not an exhaustive list. There's lots of other people as well that have helped over the years and things like that. Naturally, funding is always instrumental in any scientist being able to undertake research, and so we're incredibly grateful for the funders that have provided that, and particularly now when funding is very, very tight. So we're grateful to those, and obviously I'm grateful to the Royal Society and the Wolfson Foundation for their fellowship. And I just wanted to make a final special thanks really to 5 particular individuals that really have helped with my research over the years. That's Vince Russell from Bayer, who shaped my enthusiasm and curiosity in neutrophils. I find them fascinating, I really do, as hopefully you can tell. Rad and Marrow for their guidance through the PhD, and obviously providing a good infrastructure and setting for my research career. Neil, because he was instrumental in teaching me about the circulation, the microcirculation in particular. He was a pioneer in the use of intravital microscopy and how we can use it as a tool to understand cellular events, leukocytes, trafficking and things like that. And also Julia Buckingham. And Julia actually, although she is the VC of Brunel at the moment, it's actually because when I was at Imperial, so she recruited me first of all to Imperial, so my first academic position. So without that opportunity, without having, stepping foot on that ladder, who knows where things would be. So those are 5 people that are very, I'd like to make a special thanks to. And that's it. So any questions? Thank you very much for listening. I appreciate that. Little plug for a book. So there's some more about, if you're interested in some of the things that were spoken about, there's more about that in that book. And I'll take any questions. Thank you very much.
Well, thank you, Felicity. That was absolutely fantastic. A real tour de force there. Really great to see the history of inflammation there, 2,000 years or so of inflammation. And it's great to be reminded of those things. And it's just amazing to think as well that these processes are going in our bodies all the time, and we don't notice them. They're just happening because the system is so well controlled. So I was going to just ask a question about that. And you talked about all these inflammatory diseases, and do you think that is a problem of too much inflammation or not good resolution, or is it both that's going on? What do you think about that?
I think it's a combination, really. We do know that in chronic inflammation or in chronic inflammatory diseases that there is resolution dysfunction, and so we can measure that as I showed, in different mediators that are involved in that resolution profiling that are perhaps downregulated. And so I think in some cases there is also obviously the genetic factors that go alongside all of this that can contribute to the inflammation that's occurring, as well as I say, aging and the susceptibility to inflammatory events as well. So I think it's a combination rather than one specific thing. But the resolution, without the resolution, we're not going to restore homeostasis in that sense. Although there is still, with aging, as we know, that's inflammaging, which is a term. I have been asked that before, but it is a term. That occurs, that low-grade inflammation that occurs as we age.
Excellent. So now we've got some questions in the Q&A here, which I shall read out to you. So the first is from Pratibha Madlani, and she says, "My son is sick with lupus and suffering from lots of inflammation and fatigue. Do you suggest any anti-inflammatory diet?"
Oh, very interesting question. Thank you. Yes, I mean, first and foremost, I'd always say talk to your GP, no question about it. But we do know that diet plays an important role when it comes to inflammation and also to depression. So it has a really important role in that, and different types of foods that you can have. we know about, you know, the 5 fruits and vegetables and things like that. So those I guess vegetables that are cutaneous vegetables, so things like broccoli is very good, has an active ingredient sulforaphane, which is shown to be an antioxidant, and so that's very good for you. Omega-3s, fish oils, very good. And so again, omega-3s in the resolution profile, same profile there, so it's involved in resolution. So I'd certainly recommend that you take those. Again, I would always say, whatever you take, if you're taking supplements, then make sure that there is no interaction or with any of the drugs that that your son or whoever is taking at the time. But yes, omega-3s are definitely good, and broccoli, yes, is also very good.
Great, thank you very much. Right, another question is from Alex Sullivan. It says, I've had patients who are apprehensive to take new pharmacology to control their pain, stroke, inflammation.
Yeah.
Have yourself or any of your colleagues carried out any investigations where nutrition can have a positive or negative effect on inflammation? If not, can you reference any studies?
Nutrition,
yeah, can have a positive or negative effect on inflammation.
Yeah, so again, there are studies, certainly from the cardiovascular point of view, and Amrita Awalia is a lead on several of these studies. She's at Barts, she's actually at the William Harvey at Barts as well, showing that the effects of beetroot is very effective in cardiovascular diseases. Sulforaphane, as I've mentioned, has also been tried. It's been tried in, so this is, when I say sulforaphane, I'm talking about broccoli in particular, so that's active ingredients in broccoli or cruciferous vegetables. That's been tried, again, for patients that have undergone surgical treatments. So they gave very high dose broccoli drinks before surgery to see whether there was a protection in the possible infection that could occur because of the surgery, and certain tests were done there, and so there was a lot of positive results that came from that. Leaders in that field from the sulforaphane effects in the cardiovascular, I'd say Paul Evans, who's in Sheffield, has done a lot of work in that area. So, and Giovanni Mann at King's has also looked at sulforaphane, but in the context of stroke. We did a little bit a number of years ago, looking at the inflammation. So looking at leukocyte trafficking, and we know that if you give sulforaphane, the active ingredient of in broccoli, that you can reduce the inflammation, both in the brain as well as in other vascular beds. And we looked at that in the context of models that were stroke models as well as sepsis.
Yeah, no, thanks. And I remember seeing a presentation from Amrita Aluwalia. He talked about the William Harvey on that research, looking at beetroot juice and the protective effects it has on in vascular inflammation. But she was talking about the problems of that, because you have to drink quite a lot of beetroot juice, and it's an acquired taste, I think.
Yes, and there's always a bit of a shock when you then have to go to the toilet.
Yeah, because it comes out a different color. Yes. Okay, right, well, let's move on. Okay, so we got some questions here. So Rachel Kerslake says, your work is so fascinating. Have you conducted any work with 3D tissue growth organ on a chip to model? Oh god, the chat's just moved and now I can't see the rest of the question. Hang on. Just a second. There we are. To model these events to see if similar effects can be captured using in vitro models.
A great question. Thank you very much. Short answer, yes. Longer answer, I suppose. So we've looked at, or are in the process of looking at neurovascular models, an organ on a chip in that way. And we're doing some work actually with my postdoc, who's now an independent researcher himself, and that's Paul Holloway. He's based at Oxford, and so he's very much into the bioengineering and the biology side of things, and creating these neurovascular units, an organ on a chip. And so we're looking at that at the moment from a neurovascular point of view. Yes. So there are other members of the centre in Certin that are doing organ on a chip work at the moment. And so I would recommend you have a little look at our website. I'm quite happy to pop that in the chat afterwards, and have a look at the work that they're doing, not just within our centre, but other centres across Brunel. So there's quite a few different areas, but we're looking at it in the context of the neurovascular unit. Yes.
Great, thank you, Felicity. Now, here's another question from Sergio Cheetah. Enjoyed your talk very much. Couple of questions. One, how do you induce inflammation in your in vivo animal models? So why don't you take that one first?
Sure. Multiple ways, really. It's all dependent on the type of inflammation we want to look at. So what disease we want to look at. So if it's stroke, for example, we do a number of different models of stroke in rodents. And that's again dependent on what stroke we're modeling from the clinical perspective. So the classic one that we do is where we insert a filament. So we're inducing ischemia using a filament. And that's done not just within my lab, but multiple labs across the globe. And so in other cases, if it's acute inflammation, it might be an injection of an inflammagen. It could be, for example, I showed the video with the macrophage eating the zymosin particles. We also use zymosin as an inflammagen, and so we give an injection of that, and it can induce an acute inflammatory response. So it's different ways. Yeah, it really does depend on the model, the inflammatory model that we want to look at.
Yeah, and sometimes you can use bacteria and stuff, can't you? You can inject those to induce an inflammatory response.
Absolutely, yes. And in some cases, we've got genetically modified animals that perhaps are more, well, we use, for example, we look at sickle cell disease in my lab, and so we have mice that have the humanized form of sickle cell disease as well. So there's different models available.
Yeah, great. Okay, and then the second question from Sergio, which actually has two parts to it. So it says, questions on annexin. It was not clear if annexin is being used clinically or whether it's still being tested pre-clinically. And then the second bit is, I was also interested in what are the benefits of annexin over aspirin, for example. Can it be used preventatively? Yes.
So great questions. Answer the first one, yes. So it's not in the clinic at the moment, annexin, but it is being trialled. So so there are some results for that, but it's not got to the clinic at this stage. What was the second part?
And then the second bit, I mean, just
aspirin.
Yeah, just to finish on that bit, I'll remind you of the second bit in a minute, but just on that bit about annexin. I mean, it's a protein, so yes. You know, there are issues around, you know, lots of other aspirin's a very small molecule, isn't it? So you know, and it's cheap to produce, etc. And things like ibuprofen and steroids, you know, they're also small molecules. And I guess, you know, annexin's a protein, so it's much more difficult to make, more expensive to make, etc. So I guess that's some of the challenges around using it therapeutically, isn't it?
Yes.
You have to think about how it's administered. You know,
absolutely.
You can't take it by mouth; you have to give it by injection.
Yes, or inhalation is another option.
Oh, okay, inhalation.
So, but you're absolutely right. The cost-wise of producing a protein, and also thinking about how it can, where you want it to go, is very important when you're thinking about finding a good strategy and therapeutic target. So some of the things that we are also looking at, or other groups have done as well, is I'd mentioned that there are different portions of the annexin, of the whole protein. There are different peptide segments, and those themselves have anti-inflammatory, pro-resolving effects. In some cases, it's not perhaps as effective as the whole protein, but they're still there. And so we've looked at a lot of different models, not just us, other groups as well, have looked at being able to manipulate those peptide structures in order to be able to obtain those perhaps as potential drug strategies. To answer the question with respect to aspirin, why would you give it or why would you take it? Aspirin's an interesting one. So if you looked at aspirin today, so if it suddenly if it was developed today, it wouldn't get onto the market just because it wouldn't pass the regulations, the standards and the regulations that we now have in place in order for drugs to go onto the market. And that's primarily because it's associated or affiliated with gastro effects, so the lining often of the stomach and the effects that it has there. The idea of coating the aspirin is now a much better strategy, and obviously originally when it was first formulated, it wasn't coated. So there's a lot of can be a lot of acid reflux, esophageal complications because of aspirin. And so when you look at annexin, we don't have those side effects. We don't have a lot of those possible problems there. I'm not saying it's a better drug necessarily. I'm saying it has a different function and it works in a different way. So it has, whereas aspirin is more sort of anticoagulant, really, the annexin is able to wear different hats in different situations. We have shown it to be a strategy that you could give prophylactically, so you can prevent the effects or the negative effects, let's say, of stroke. So we've given it before we've given animals a stroke, and we've shown that the effects of that stroke are far reduced if that animal or animals have taken annexin beforehand. Yeah. So there's a lot of work still going on, and obviously, as you say, Paul, you know, it is a cost benefit as well for certain drugs, and the making of it as well. It's not an easy thing necessarily to make.
That's right. That's right. Okay, so we got there's another question here from Mabel Akizi. I hope I've got that right. Thank you, Professor Gavins, for the lecture. From your research and experience, why is surgery, not just medication alone, used for some cases? She gives an example saying programs like Meet the Elephant Man. I'm not quite sure where that's coming from, but I guess sometimes, you know, surgery has been used in things like TB, hasn't it? You sort of do lung resection because the immune response there, I guess, is so large that you know it's the only way of dealing with things. You have to sort of take the organ away because, you know, drugs wouldn't treat it. But I guess there may be some examples in, you know, I guess when it's severe. I mean, you know, if you have a really severe inflammatory response, you might get things like and thrombosis. You might get things like gangrene occurring, wouldn't you?
Absolutely.
You know, in the foot. So you have to amputate it, and the drugs and anti-inflammatory drugs or anti-thrombotic drugs won't do anything because it's too late.
And it's dependent on what comorbidities that patient have. You know, so a lot of diabetic. Well, I say a lot. Some diabetic patients, you know, have that. They have a lot of inflammation that's there. And yes, you're absolutely right that providing or taking anti-inflammatory drugs isn't sufficient. You know, once you have got gangrene there, there's nothing you can do really from that point of view. You can reduce the after effects from that, you know, but amputation is unfortunately the treatment.
Yeah.
Okay.
Okay. So we've got another question here from Pam Holloway. Do immune suppressants affect inflammation?
Yes, they can do. Yes, it's dependent again on which specific drug, but yes, they do. They can have quite a profound effect on inflammation. Yes. And
I guess that's why there's problems with, you know, immunosuppressants can then make people susceptible to infection, of course. So you have to be very, very careful. And I think we've seen that actually during COVID, you know, lockdown period, people have, you know, had to be, you know, shielding people who are taking immunosuppressants because of the high risk in getting infected. So yes, they do, very much so. Okay, well, there are no more questions, so I think we'll draw this to a close. So I'd like to thank Felicity very much on behalf of everybody. Thank you very much for just a fantastic presentation and for answering all our questions as well. I'd like to thank the audience for attending. I'd like to thank colleagues for Tom and Seb for being behind the scenes here and making sure it ran so smoothly. So well done. And please look out for future events as part of our research festival series. There's some more open lectures happening over the next few weeks. So please enjoy those and, you know, register for those online. And thank you again, Felicity.
It's a pleasure.
Bye-bye.
Cool. Thanks, everyone. Take care.